In people who live past 110, close to 1 in 5 of their T cells belong to a rare type that almost nobody else carries in those numbers. Researchers at the University of Osaka drew blood from 28 adults split into 3 age groups, from 70 up into the supercentenarian range, and found a clear staircase. 4% of a certain immune cell in the youngest group, 9.6% in centenarians, 17.6% in people over 110. The cell is called a CD4 cytotoxic T lymphocyte, a killer cell that normally shows up to fight tumors and chronic infections. Headlines have already turned this into a longevity secret, a hidden reason some people simply outlast everyone else. The actual paper, published in Cell Reports, is more careful than that framing, and more interesting. Today you get what the researchers measured, what these cells appear to do, why the same finding could just as easily be read as a warning sign as a superpower, and the honest limit of drawing a longevity lesson from 28 people. The team, led by researcher Kosuke Hashimoto, sequenced immune cells from adults in 3 brackets, ages 70 to 99, 100 to 109, and 110 and older. The last group known formally as supercentenarians, a population so rare that most countries can count their living members on 2 hands. Across every bracket, the proportion of CD4 cytotoxic T lymphocytes rose in step with age. And inside the oldest group, those cells were not just more numerous. They were more concentrated. The single largest immune cell clone in a person's blood made up an average of 1/3 of all their CD4 cytotoxic T cells. In one centenarian, a single clone accounted for more than half of them. That is a strikingly narrow immune signature for cells whose entire job is broad surveillance, hunting down abnormal cells wherever they appear in the body. The researchers describe this as evidence that the immune system does not simply wear down with extreme age. It reorganizes, concentrating its resources into fewer, more specialized fighters. The detail that got the study attention outside academic circles involves cancer. When researchers compared the receptor sequences on these supercentenarians' killer cells against public databases, nearly 3 dozen matched sequences previously found in cancer patients, in lung, breast, and liver tumors specifically. None of the supercentenarians in the study had those cancers. One reading of that overlap is reassuring. These people may be carrying a standing immune force already primed to recognize and destroy a kind of abnormal cells that turn into tumors, built up over decades of encountering and defeating small threats before they became visible disease. Hashimoto put it plainly, saying immune aging is not simply a process of decline, and that even in extreme old age the immune system may keep adapting to new challenges rather than gradually giving up. That reframes a population usually discussed only in terms of frailty. Reaching 110 may not mean an immune system that limped across the finish line. It may mean one still actively working. Here is the question the study cannot answer on its own. Does having more of these cells help a person live to 110? Or does living to 110 simply produce more of these cells as a side effect of decades spent fighting off infections and abnormal cells the ordinary way? Those are very different stories wearing the same set of numbers. It is entirely possible that the immune systems of supercentenarians look this way because surviving 11 decades of exposure to viruses, injuries, and cellular damage naturally selects for these particular killer cells over time. The way a well-used tool sharpens rather than dulls. Under that explanation, the cells are a consequence of a long healthy life, not a cause of one. A narrow, Dominant cell clone could also reflect a different kind of story entirely. An immune system that has run out of diversity and is repeating itself because it has fewer new responses left to make. The Osaka team gathered a snapshot, not a timeline. And a snapshot cannot show which direction the arrow of cause runs. 28 people is a small number for a finding this widely shared. It is enough to establish a real, measurable pattern worth publishing and worth investigating further. And it is nowhere near enough to tell you what these cells would do if boosted, transplanted, or triggered on purpose in someone who is not already a naturally long-lived supercentenarian. The researchers say this themselves in the paper. Their data do not establish that high levels of these cells protect against cancer or directly extend life. And they still do not know what these cells are doing elsewhere in the body beyond the blood samples they measured. There is also a less comfortable possible reading sitting right next to the hopeful one. A single clone dominating 1/3, or in one case more than half, of a cell population can be a sign of a healthy focused immune response. Or it can be an early signature of clonal blood conditions that become more common with extreme age, and are not automatically benign. The study was not designed to tell those two possibilities apart. And nobody honest about the data claims otherwise. This one splits down the middle. The signal is real. Supercentenarians consistently carry a distinctive, dominant population of CD4 cytotoxic T lymphocytes that ordinary aging adults do not. Verified across 28 sequenced individuals in a peer-reviewed study. And the researcher's description of an immune system that reorganizes rather than simply declines is a genuinely useful correction to how most people picture aging. The noise is everything built on top of that finding. The idea that scientists have located the secret ingredient of extreme longevity. That this cell type causes long life rather than merely accompanying it. Or that the finding says anything yet about intervening in a healthy body to chase the same result. The researchers who ran the study were explicit that they have not shown cause and effect. And a sample of 28 people cannot carry a conclusion that large. What survives is a genuinely interesting biological clue about aging bodies, not a longevity formula. Subscribe for the science headlines that get separated from what the actual data proves. Signal from the noise. Every week.